So, a brief recap of the light reactions. Okay, so here we have our thylakoid membrane. Okay, and the light reactions happen in that thylakoid. Photosystem 2, electron transport chain to photosystem 1. Notice here in photosystem 2, that electron transport chain, very similar to cellular respiration, produces an electrochemical gradient of hydrogen ions here inside the thylakoid that uses that same proton motive force to spin ATP synthase and make a great deal of ATP. Okay. Photosystem 1 now uses that electron to charge up the NADP, okay, NADP plus, to an electron carrier that's NADPH. And now we're going to use both this ATP and NADPH to produce the glucose that fuels this planet. Okay. Let's take a look at the next step now, which is the Calvin cycle. Okay. So the Calvin cycle is the opposite of the citric acid cycle or the Krebs cycle. Okay. So just like we said before, the cellular respiration and photosynthesis are opposites of one another. So where cellular respiration had glycolysis, Krebs cycle, and electron transport, photosynthesis is the opposite. We had electron transport first through the photosystems. Now we're going to have a cycle, in this case, Calvin cycle. And the last thing we're going to do is literally put together glucose, the opposite of glycolysis. So let's take a look at the Calvin cycle. This is a three-phase cyclical set of chemical reactions. Okay. Orient ourselves. The light reactions happen inside the thylakoid. Okay. And the Calvin cycle now happens out in the stroma. Okay. So if this is a stack of pancakes, think of that stroma as a sweet syrup, because that's where the sugar is going to be produced. So here we have this is a Calvin cycle. Here we have incoming 3 molecules of carbon dioxide from the atmosphere. We've got 3 stages to this cycle. Number 1 is what's called carbon fixation. Okay? Fixation is a generalized chemical biological term that describes taking something from the air and causing it to be a part of a biological molecule. Okay? Nitrogen fixation is a common issue in agriculture. How do we get nitrogen back into the soil? How do we get nitrogen to our plants? Okay, so carbon fixation, similar concept. Okay, stage 2 is reduction. Okay, we're going to reduce that carbon dioxide, okay, forming smaller sugar molecules. The third stage here is regeneration of the molecule that starts this whole thing. Okay, the molecule that is able to combine with carbon dioxide from the atmosphere has to start this whole Calvin cycle over and over again. Okay, so carbon fixation, reduction, and regeneration. We'll take a closer look at that. Okay, so phase 1, fixation. Okay, we have carbon dioxide combining with a molecule that's abbreviated RUBP, okay, the ribulose bisphosphate. This is the molecule that starts it and ends it. Okay. ATP then comes in, is used. Okay, our NADPH is also used to reduce those molecules to what's referred to as G3P. More to follow on G3P later, but this is our base sugar. Okay, this is our base carbohydrate at this point. Okay, after the reduction phase, we are going to go through regeneration of RUBP so that we can grab more carbon dioxide out of the atmosphere. This is the Calvin cycle.